His research His research focuses on how sensory motor signals contribute to metacognition, and today he's going to tell us about single unit recordings in humans and what they revealed about the relationship between perceptual consciousness and metacognition. So, Nathan, if you're ready, take it away.
I am ready. Thank you, Nadine. Thank you for this nice introduction, and thank you also, Nadine and Steve, for this kind invitation. I'm pretty happy to take part in this distinguished club. I was telling Steve just before we started that I felt a bit rusty. I haven't given talks in a while with the current situation, so I hope this will go well. My goal today is to present you some recent results we obtained indeed about single unit recordings in humans, but not only. And I'd like to propose a shared mechanism for perceptual consciousness and perceptual monitoring, which involves evidence accumulation. So to start off, I'd like to give some pretty basic definitions of what we're talking about. So not to make a consensus, but just to make sure that we are on the same page when interpreting results. And I will start by defining perceptual consciousness simply by saying that we consider this as what it is like to see, feel, hear, smell, or touch. So the subjective experience, the phenomenal experience, for instance, associated with this person here looking outside to a man walking his dog. So one can imagine that there is indeed something it feels like to see a dog. Okay, so that's for perceptual consciousness. And today I will also be speaking about perceptual monitoring, which this time I define about our capacity to think about our own thinking. So our capacity to introspect and, for instance, to judge the quality of any given perceptual representation we may have formed. And all the work I'll be presenting should be situated within the quest for the neural correlates of consciousness that was started by Crick and Koch and others some decades ago. And here's the main situation. So we're still considering this man looking outside to this guy walking his dog. And the game here in this quest is to identify in this observer's brain a set of neurons that we call the neural correlates of consciousness, or NCCs, which are minimally sufficient for a subjective experience to occur. For instance, here, the subjective experience of a dog. Okay. And so the game is to separate these neurons from all other neurons that do a bunch of stuff, but that is not directly related to subjective experience. And we typically do so using a contrastive approach that was proposed by Bernard Bars in the 90s, where we compare brain activity and behavior in case a stimulus evokes a subjective experience, so the percept of a dog, to the same stimulus when it doesn't evoke any subjective experience at all. And so by contrasting brain activity with and without subjective experience, one can isolate, given some typical confounds I ruled out, the neural correlates of perceptual consciousness. Now today I would like to extend this approach to another level, which is this time trying to document some neurons that are involved not in the subjective experience of the dog, but of our capacity to introspect and monitor how faithfully we represent that dog. I don't know if this is on your way or not. I see my face instead of the dog. Okay, so the way we do this, the way we study perceptual monitoring in the brain is typically through confidence ratings. So we ask a subject to report a percept and then indicate how confident he or she is about his or her response. And so we speak of the calibration of confidence, that is the capacity to adapt confidence to one's own performance. And I won't tell you much new here today. Steve and his colleagues have done a myriad of brilliant studies in the last decade studying this. Okay, that's for the neural correlates of consciousness. Now, as I said in the first slide, today I want to bring a mechanism to this NCC, which is evidence accumulation. So that's a very broad concept that is used in many domains, including neuroscience. And typically, we define this as a process by which noisy sensory information is sequentially sampled until sufficient evidence has accrued to favor one decision over others. So that's, again, a very broad concept that has been recently discussed in the concept of consciousness. And for instance, Stan Dehaene, among others, has proposed within the global workspace theory of consciousness that evidence accumulation following the processing of a stimulus increases up to a level which may serve as a trigger for what he calls ignition. So this widespread activity throughout the brain that is associated with conscious access. Okay, so evidence accumulation would be like a necessary step before ignition takes place, and therefore before conscious access occurs. That is at the theoretical level. Now, if we look at things more practically, how it's done empirically, we typically use these kind of models where we assume that, so there's a stimulus presented to a subject, and we model evidence in the brain as following like a noisy trajectory, but accumulating over time. Here, the horizontal axis is time, until it reaches a threshold. And depending on the rate with which this evidence accumulates over time, with a concept we call, or parameter we call the drift rate, sometimes this drift is a bit lower, so evidence accumulates slowly, and meaning that the decision bound is crossed at a later time. So with this, we can account for decisions being taken at different moments in time. And sometimes we can assume that evidence is not sufficient and the decision bound is not crossed at all, which would result in no decision. I should say that these models have been used more predominantly for discrimination tasks, and there's not much work studying this in detection tasks, which are most relevant for consciousness. The goal here of these models, important to remind us, is that we want to fit distributions of reaction time. So we want to find a set of parameters that generate those trajectories, which in turn result in let's say, a gamma distribution of reaction times that we would observe. And a big problem here is that, of course, in case there is no response, there is no reaction time to model. So maybe that's a reason why there's a bit of a missing link here between what's done empirically and claims at the theoretical level that assume that evidence accumulation precedes conscious access. So how do we measure evidence accumulation in the brain? There are several signatures that have been described, mostly from non-human primates. And today I will speak about two main signatures of evidence accumulation. One is that accumulation should be indexed by a neural process that varies according to the response time. And another signature is that this accumulation process should be indexed by a process that varies according to stimulus intensity. So here I'm showing you an example of such signature that was described by Connell and Simon Kelly and others with EEG. So subjects were asked to detect a target while we measured EEG. And what you can see, if you look at electrodes, parietal electrodes, as you can see here, the topography, is that you can find an EEG component, which is called the CPP, the central posterior positivity, with an amplitude pattern that increases as a function of response time. So you can see that the EEG amplitude increases simultaneously for in all cases, but it reaches a peak that occurs more quickly for fast responses, a bit later for medium responses, and even later for slow responses. So when we observe such pattern, we can assume that evidence accumulation is involved in the process. And same thing occurs for stimulus intensity. If you measure the same CPP, so as you can see, the topography is very similar, you can find that the amplitude starts rising at the same moment, but reaches a peak earlier for strong stimuli than for weak stimuli. So these are two indirect ways to prove that there is evidence accumulation occurring, and it's really important to remember, because I will be using those two signatures later in the talk. So evidence accumulation has been used not only to account for first order decisions, but also for confidence ratings, and therefore to study perceptual monitoring. There are two main types of models that have been proposed. A first one, here I'm showing you a classic model by Pleskak and Busemeyer, which proposed a way to model confidence with these diffusion processes by allowing a second stage diffusion. So after the decision bound is crossed, these two authors assume that diffusion could still continue, and they use the second diffusion stage to model confidence in one's decision. You can also model confidence with another family of accumulators. This time you can, for instance, a paper by Vandenberg in eLife in 2016, assuming that you're presented with a stimulus that can move rightward or leftward. You can model confidence by assuming that there exists two accumulators, one corresponding to the evidence in favor of a right motion, another accumulator for the left motion, and you can define confidence by letting these two accumulators diffuse and take the ratio of these two accumulators as a proxy for confidence. Using this family of model is a bit tricky in case of a detection task, because it's pretty hard to imagine that you would accumulate evidence in favor of an absence of a stimulus. So later on, I will mostly be speaking about this family of evidence accumulation models for confidence. All right, so our goals today are multiple. So first, I'd like to propose a measure of evidence accumulation in a contrastive study of consciousness, comparing situations where a stimulus evokes conscious experience versus when it does not. Considering at the same time detection responses and confidence reports, while ruling out some typical confounds that have hindered the definition, the definition of NCCs. And typically, I'll be speaking about motor confounds or confounds in terms of task demand. And finally, I will show you some neuronal implementation of this mechanism. This work has been submitted recently on bioRxiv. This was led by Michael Pereira, who you can see here. And I should take a second to publicly thank him and acknowledge the tremendous work he's done in this article. It's mostly his stuff that I'm presenting here today. And I should also say that I have a pretty spotty connection these days. So in case I suddenly disappear in the void, I asked Michael, who kindly accepted to take over for a few minutes. So he's really the man of the situation today. Okay, so I will present you today 3 different data sets, starting with a data set from an epilepsy patient we were lucky to collaborate with. It's a pretty rare patient for 2 main reasons. The first reason is that he was implanted with a microelectrode array. So that's this little chip that you can see here that is smaller than my fingernail. These are rare, and even more so because it was implanted this time on a region that is quite rarely implanted normally, which is the parietal posterior cortex. Next, I will present you a data set with healthy volunteers involving this time EEG and computational modeling. And finally, another population of patients which undergo surgery for deep brain stimulation. All right, so I'll start with the epilepsy patient. That's a project we conducted in Geneva in collaboration with the VIS Center. And again, we had the chance to work with this young patient who was 23 years old. He was cognitively intact, and he had intractable epilepsy. To document the origin of his seizures, he received a grid of electrocorticography electrodes that you can see here. On the left parietal cortex, covering precentral regions and up to like here posterior parietal cortex. And as part of a research program, he also received this microelectrode array. So this little chip which possesses 100 channels that can measure single neurons. Okay, and as you can see, the microelectrode array was located right on the bank of the postcentral sulcus in a region that I will call the posterior parietal cortex, which is known to be a hotspot for evidence accumulation. So when we knew this patient was programmed, we're pretty excited, and this guy was very keen in participating in our experiments. So we did 3 experiments with him. One in which he had to report immediately what a stimulus that he detected. Another one where we imposed a delay between the stimulus and his responses. And finally, a third experiment where he had nothing to do and no report experiment. So this is our paradigm. Because we had electrodes in sensory motor regions, we opted for tactile stimuli instead of the classic visual stimuli that we're more familiar with. So we equipped our patient with a little vibrator here on the right wrist. And we could control this vibrator with a sound card and send vibrotactile stimuli with a very fine-tuned intensity and duration. So we made sure to send a stimulus with an intensity that would correspond to about a hit rate of about 50%. So half the stimuli were detected and the other half were not detected. We used for that a staircase procedure. And the task of the subject was simply to press a key as soon as he felt something. Okay. So if a key press occurred within a 2 seconds window following stimulus onset, we define this as a hit in blue. And if a key press did not occur within these 2 seconds, we considered this as a miss. And in case the patient pressed the key outside the stimulation window, we considered this as a false alarm, which happened only once or twice in this experiment. I should say that we made sure that stimulus intensity or stimulus onset did not vary between hits and misses to make sure that at least in terms of stimulus feature, there was no obvious difference that could explain our results. Here I show you the distribution of response times that we obtained from this patient. And as you can see, when the patient felt a stimulus, he pressed less than 1 second on average after stimulus onset, which means that we do not mix up hits and misses here. There's quite a clear separation between the two stimulus categories. So next, I will show you the data from this microelectrode array. So we end up with a time series from this microelectrode array that we submit to a spike sorting algorithm that allows us to detect individual neurons. And in this experiment, we could isolate 186 neurons, among which 81 had different firing rates between hits and misses. And we made sure with permutation tests that we could not observe this just by chance. And most of these neurons had a higher firing rate for hits than for misses, although we found a few neurons that also encoded specifically misses. So among these 81 neurons, 14 of them showed a correlation between spike rate and response time, which is pretty interesting because it's a signature of evidence accumulation, as I said earlier. And I will show you now 2 neurons among these 14. So that's one example and another example here in the right column. The top row describes the waveform, so the average spike of each of these neurons. Below you have a raster plot where, so you have time on the horizontal axis, 0 is the stimulus onset. And here on the y-axis, you have trials that are reordered as a function of the response time. So in case of a slow response, medium response, fast response, or no response at all in red in case of a miss. And each little dot is a spike that we measured. So now if you aggregate those responses on the y-axis, you obtain the plot at the bottom, which represents the firing rate, okay, over time. So as you can see, after stimulus onset, you have an increase of firing rate that is stronger for hits than for misses, but that also occurs earlier for fast hits versus medium hits versus slow hits. And again, that's considered as a signature for evidence accumulation. We also analyzed the ECOG grid, and we looked for this time electrodes that had local field potentials whose amplitude differed between hits and misses. As you can see here, the column map indicates how much this electrode distinguished hits from misses. You can see a widespread effect, both in premotor regions, but also in more posterior regions. And the amplitude of these LFPs also showed this hallmark of evidence accumulation with a steeper and earlier increase of local field potentials for fast responses versus slow responses. Okay, so that's pretty cool. In terms of evidence accumulation, we have evidence at the single neuron level in humans, which is, to the best of our knowledge, a first. But to study consciousness, it's not so interesting because there's an obvious confound here, which is that when the subject felt something, he pressed a key, whereas in case of a miss, there was no key press. So it's possible that we're documenting NCC, but it's also very possible that we are documenting motor confound between hits and misses. That's why we're turned to experiment 2, which was pretty similar to experiment 1, but had a few additions. So first of all, we added a temporal delay between the stimulation window and the response, so that anything we measured in the brain around stimulus onset could not be contaminated by the act of responding. We also added 20% of trials with no stimulus at all, which allowed us to measure correct rejections. And this time the response was provided verbally instead of motor key press to make sure that our effects would generalize to other response effectors. And last but not least, we added a confidence rating. So after each trial, the subject had to tell us whether he was sure or unsure about his detection response. Here is the behavioral result from this patient. So we have miss, hits, and correct rejections. Again, we made sure that stimulus intensity, stimulus onset was equivalent in all categories. Here you have the distribution of confidence for hits and misses, and you can see that overall misses were associated with a lower confidence than hits. And because we had a few trials in this clinical setting, we just combined together ratings 1 and 2 as low confidence and rating of 3 as high confidence. Now, in terms of neural data, this time we identified 86 neurons, which we analyzed with a factorial strategy. So we tried to explain the spike rate of these neurons as a function of both detection and confidence. We found no main effect of detection. So only one neuron had a firing rate that varied only as a function of detection. And those permutation analysis we did reveal that we could have observed this by chance. Same thing for confidence. But we found 70 neurons with an interaction between detection and confidence, and this could not have been observed by chance. Here again are two examples of such neurons. So waveform, raster plot, and I will focus here on the average firing rate. And what you can see here is that there is a specific increase in this dark blue curve, which corresponds to high confidence hits, whereas low confidence hits and misses were virtually indistinguishable at the single neuron level. Okay, so that means that taking one neuron at a time, we couldn't make the difference between a miss and a low confidence hit. Now, that doesn't mean that at the population level, nothing happens. And it could well be that single neurons do not encode this difference, but as a population, this occurs. So to address this, what we did is we considered all neurons together and fed them to a decoder, which was a simple weighted average of the neuronal input. And with this decoder, we built an algorithm that was able to distinguish hits from misses. Okay, so it's obvious that we managed to do that because taking individual neurons, we could do this. But what was interesting is that taking the output of this decoder, we could find that the decoder output correlated with confidence for hits that is described here. That means that there is information relevant to confidence in a decoder trained on detection, suggesting that there may be an overlap between the representation of detection and confidence in these neurons, but taken as a population. I should also say that these experiments, experiment 1 and 2, were run on different days. So we're pretty much running between the hospital and the lab and designing experiments on the go. And unfortunately, we cannot make comparisons between the neurons we found here and the neurons from experiment 1, because there's no way we can make sure that they are similar. Some die, some we do not record anymore, some new appear. So it's difficult to make a link between those two sets of neurons. We also analyzed the ECOG grid. Same thing, looking for an interaction between detection and confidence, this time in the amplitude of local field potentials. And again, if you look at the color map here of each electrode, you can see that the significant electrodes this time are all, well, it's only 2 of them. They are situated in the posterior parietal cortex. And the effects we found in experiment 1 in anterior regions, motor regions, disappeared, meaning that probably there was indeed some motor confounding in experiment 1. And so in these 2 electrodes here that really are near to the microelectrode array that is represented in green, we found the same pattern of results with the amplitude of LFPs showing a significant increase for high confidence hits, but no big difference between misses and low confidence hits. Okay, that's pretty nice. We ruled out obvious confounds in terms of motor button presses, but there is another possible confound, which is task demand. So as you may know, there's an ongoing debate right now, and we wonder if to study consciousness per se, we should not be doing it without asking the subject to do a task at all. So in that case, the subject, after feeding a stimulus, he had to maintain the representation in his working memory, then prepare a verbal response, then prepare a confidence response. So we don't really know whether what we measure here is simply related to subjective experience of feeling something vibrating on your wrist, or is it related to the task that is associated with the stimulus. So are we measuring a neural correlate of task or a neural correlate of consciousness? So we went back to the hospital again, but this time we gave a break to our poor patient, and we simply equipped him with a vibrator and let him mind wander while we sent him either low vibration, medium vibration, or strong vibrations. Okay, so he had nothing to do, just experience the vibration when possible. And we checked if we still measured neural activity that would encode the stimulus in the absence of a task. That's what I show you here. So we identified 96 neurons. And among these 96, 14 of them showed a main effect of intensity, meaning that the mean firing rate increased more so for strong stimuli than for medium or weak stimuli. Which, if you remember, the introduction is another signature for evidence accumulation, this time that we observe without a task. And this pattern is very similar to the pattern of result I just showed you from experiment 2. Meaning that this correlate that we found does not disappear and is not only attributed to the task at play. Okay, so time for some interim conclusions. We described some PPC neurons with a spike rate that is proportional to response time and intensity, which are two hallmarks of evidence accumulation in humans. We found neural correlates of detection and confidence in the posterior parietal cortex, which are independent of response, effector, and task demand. So after we tested this patient again, went back to the lab, and we were pretty excited by the results and the findings, but at the same time, we're a bit worried about the generality of these findings and how we would publish them, given that it was only one single patient. We looked at a very, very specific patch of tissue in the parietal cortex, very small. So we measured 100 neurons among the billions available, with a very limited number of trials in a clinical setting, you know, with a nurse that can come in and come out at any moment. So it's a beautiful set of results, but we were a bit worried that in fact we might have been missing the forest for the trees. So how can we generalize our findings to a broader sample? So we came back to good old EEG and good old healthy volunteers, psychology students who come to the lab and perform the same experiment with much more trials. This is the experiment we conducted next. So an EEG experiment on healthy volunteers, 18 of them, very similar to the experiment done by the patient, except this time we had 500 trials per subjects, which allowed us to not only describe the behavior better, but also use this time visual analog confidence scale, which allowed us for more refined analyses. So here I show you a summary of the behavioral results. On the x-axis, you have number of trials for hits, misses, correct rejections, and false alarms. As you can see, there's about the same number of trials for hits and misses, about 20% of correct rejections, and very few, again, false alarms, which we could not analyze here. And as before, we made sure that the stimulus intensity and stimulus onset did not vary between hits and misses. We obtained those nice distributions for confidence ratings in each category. And just to be brief about it, so what we found is that when a subject responded yes, so in case of a hit or a false alarm, we observed higher confidence for a correct response, so for a hit. And same thing occurred when the subject said no, so in case of a miss or correct rejection, but again, confidence was higher for a correct rejection, meaning that there was some metacognitive monitoring happening here, even though it was weaker for no responses, which has been described previously by Matt Mazur and Steve and others. Okay, now I will turn to the EEG results. So same thing, we followed the same approach as for the single unit patient. We analyzed the EEG amplitude with a factorial analysis, looking for an interaction between detection and confidence, which we found here in a cluster of electrodes on the corresponding to posterior parietal scalp regions. And if you take this cluster and you plot the amplitude over time, you obtain this ERP, where you see, again, pretty similar to the results before, that there is an increase of amplitude for high confidence hits that is much bigger than for low confidence hits and bigger than for misses. And again, we tried to see if there was some overlapping representation between detection and confidence in the EEG signal. So following the same approach, we took all EEG channels, fed them to a decoder that took a weighted average of these EEG amplitudes and allowed us to decode hits from misses. That's what I am presenting you here. So this is the decoding performance over time from stimulus onset. And you can see that after 200 milliseconds or so, the decoder is able to distinguish hits from misses. And now, if we take the decoder outputs and correlate it with confidence ratings, we found a region that is outlined here with a significant correlation, meaning that during this period of time, there is some overlapping EEG representation of both detection and confidence, suggesting that they might share a neural representation. Okay, since we had a ton of trials, we could attempt to fit computational models, and I will go back here to the good old drift diffusion model. One obvious problem here that we have is that we have no meaningful response time to fit. Because we impose the delay between the stimulus and the response, any response time we measure is not very interesting. We don't have a good proxy for the moment at which the subject took his decision instead. So to fit such model, Michael had a great idea, was to, instead of fitting a gamma distribution of reaction times, he proposed to fit the shape of the EEG response, which is what I will show you now. And it's relevant because the shape of the EEG response does not vary even if we impose a delay between the stimulus and the response. So that's our model. It's a classic drift diffusion models with some parameters that some of you may recognize, and an additional parameter which is quite important. So we consider a decision variable that oscillates. It's a stochastic process, so there is noise in the signal. It oscillates, but from the moment the stimulus is presented, there is a boost in drift rate, and therefore the decision variable increases up to crossing a decision bound. And if this thing happens, we consider that the stimulus was detected and it's a hit. Okay. So we fitted a drift rate for each subject, a decision bound for each subject, so that we can account for some people that are more conservative than others. And we also fitted an important parameter, which is a leakage parameter, which drives the decision variable back to zero constantly. We did this for two things. First of all, this allowed us to prevent noise to be accumulated in the absence of a stimulus. So if you have no leakage, you would accumulate noise and reach the decision bound all the time, and resulting in false alarms that we don't observe in the data. And also this leakage factor allows us to drive back this decision variable towards zero after it reaches the threshold, which is necessary if you want to mimic a typical EEG response, as I showed you earlier. In case of a miss, the same thing occurred, except that the drift rate was not sufficient so that the decision bound is not crossed, and therefore we define this trial as a miss. And in case of a correct projection, there is no stimulus presented at all. So due to leakage, you don't accumulate noise, and the subject responds, no, there was no stimulus presented. So we fitted all these parameters and some others that I don't show you now to the behavioral data and to the EEG data to distinguish hits from misses from correct rejections. But we also fitted it to confidence data. For that, we tested different means of defining confidence. And for the sake of time, I will just present you the rule that worked best. It's quite a simple rule. It just defines confidence as the distance between the decision bound and the maximum of evidence accumulated within a trial. So for this trial, for this hit, that would be this distance, and for this miss, that would be this distance here. So this simple rule is pretty nice because it can account, for instance, for misses in which you have a high confidence. So for instance, if you don't accumulate much evidence at all, the distance between the bound and the max of this low maximum of evidence accumulated is big. So that would result in a high confidence miss. And for instance, if you keep accumulating evidence after the decision is bound, is crossed, sorry, you would end up with a high confidence hit. Now we'll present you some of the model fits. The data is represented by circles and the model fits by crosses. As you can see, we could reproduce fairly well the hit rate and false alarm rate we observed with our EEG 18 subjects, sorry. We could also reproduce the mean confidence for hits, misses, and correct rejections, and also the distribution of these confidence ratings. And finally, we could reproduce the shape of the EEG responses corresponding to hits and misses. So we are pretty excited by this novel fitting procedure. We think that this leakage factor is interesting because it allows fitting EEG data even in the absence of behavioral response or when the timing of these responses is irrelevant, as occurs in our case. So it's a good tool to fit detection tasks. And for confidence, this novel fitting procedure assumes the existence of post-decisional accumulation, which is a natural way to mimic the metacumulative noise that has been described in many studies before ours. And it provides two simple definitions of detection and confidence. It defines detection as the thresholded accumulated evidence in time and confidence as the max of accumulated evidence in time. Third and last data set, we switch gear and we go to a complete different population, complete different brain structure. This time I will present you some results about patients undergoing surgery for deep brain stimulation in brain structure that is the subthalamic nucleus. And you may wonder, why the hell do we study the subthalamic nucleus when we deal with tactile detection and confidence? Well, in fact, it's been described in many interesting papers that the STN might in fact regulate the decision threshold through interactions with the cortex. So there are many papers during discrimination task or go, no-go task, conflicting tasks, which propose that the STN serves as a modulator of this decision bound. It pushes it up. For instance, when there is a conflict and you need more evidence to make a decision, or when instructions favor accuracy over speed, the role of the STN could be to push this boundary up. So we thought that the STN might also be involved in this model, but this time applied to detection and confidence report. And we can study this in two different ways, either based on correlational evidence, where we simply measure the STN for hits, misses, and high and low confidence. And because it's a very deep structure, we need to rely on intraoperative recordings. And we can also do so through causal evidence, where we either lesion the STN in case, for instance, of a subthalamotomy, or more recently, when we inhibit the STN through deep brain stimulation. So we're involved in the two strategies, let's say, but today we'll only speak about the correlational evidence. Okay, so we collaborate with neurosurgeons from West Virginia University in Morgantown in the US, who on a very regular basis implant electrodes in patients with Parkinson's disease or essential tremor. And the goal here is to use a little pacemaker that you can see here to stimulate the STN, inhibit it, and alleviate some of the motor symptoms in these patients. During the surgery, to make sure that the neurosurgeon is in the right spot in the brain, he inserts a microelectrode that is not used to stimulate, but that is used to record single neuron activity. Okay, so the patient is awake, and after the microelectrode is inserted, the neurosurgeon gives us 20 minutes of research time where we can actually perform the experiments I told you earlier about. So this is us freaking out, trying to run our experiment and hoping our code won't break while the neurosurgeon waits for us. The patient just woke up from general anesthesia, and we ask him or her to detect those weak vibrotactile stimuli and tell us how confident he or she is in the response. This is preliminary data. Among 11 patients, we isolated 19 neurons, a quarter of them being showing a different spike rate between hits and misses. Here I show you two examples. In both examples, you can see that after around 80 milliseconds following stimulus onset, you have a sharp increase in spike rates, but more so in misses than for hits. So this is the complete different picture as the one I presented you earlier. This time misses evoke a stronger evoked response, which goes well with the idea that when the STN spikes more, it pushes the decision bound up, and this results in more misses than hits. If you take the same neurons and then you split the response this time as a function of confidence, you see that both for hits on the left and for misses on the right, you have higher spike rate for low confidence than high confidence ratings. So we're still figuring out a way to feed these results into our models, but we have a hunch that a modulation of decision bound would also result in more low confidence rounds. So this is a very long lasting and a difficult experiment to run. We've been on it for, I think, 2 or 3 years. We're still at the stage of preliminary evidence because we have a few neurons, but these results could suggest that the STN is involved also in detection and confidence. And at the moment, we are also trying to assess the causal role of the STN with deep brain stimulation, this time collaborating with OCD patients. And we try to see if by stimulating the STN through DBS, we could lower this decision bound by inhibiting it, and therefore resulting in a more liberal detection and confidence ratings. I'm done with the data. I hope you're still with me. I just have a few take-home messages before I finish. So regarding perceptual consciousness, I like this definition of detection as the thresholded accumulated evidence that we documented irrespective of response effectors or task demands. It has a series of theoretical implications maybe we could discuss. First of all, it considers conscious access as a all or none phenomenon, which has been proposed many times, but I think this is a nice empirical validation of this proposal. And of course, it would be important to verify that this also occurs for maybe more complex stimuli. It's maybe not so surprising that a simple vibration is perceived as all or none. So it'd be interesting to see if more like multi-feature stimuli also involve all or none access. It could support prediction made by the global neuronal workspace theory, saying that crossing the decision bound may trigger ignition. So now we'd like to perform experiments with different recording sites where we could see what happens after the decision bound is crossed as instantiated by the posterior parietal cortex. It'd be nice, for instance, to see what occurs in prefrontal regions. But of course, there's a necessity versus sufficiency issue here. By no means I want to claim that evidence accumulation is both necessary and sufficient for consciousness. We know that it occurs in many cognitive functions. It also occurs in organisms such as C. elegans or zebrafish, for which we're not sure whether conscious access is similar to ours. And it also occurs, in fact, in unconscious humans. So it will be important in the future to test if how specific this evidence accumulation is to consciousness. And the last series of take-home messages for regarding this time perceptual monitoring. So we define confidence as the max of accumulated evidence in time, which can nicely account for how we monitor the absence of a percept, for instance, in case of misses and correct rejections. And we think that this leaky accumulator model we propose, with the existence of an accumulation that persists even after the decision is made, can account for some interesting features of confidence and metacognition. And finally, I'd like to thank you all for listening to me. I'd like to thank those amazing patients we worked with. I think they should be commended for taking those pretty boring experiments at times, at difficult times in their life, either during a brain surgery or just after a brain surgery. And special thanks to Michael Pereira for his amazing support, and all my other colleagues. And finally, if any of this made sense to you, there would be open positions, postdoc and PhD students available soon in my lab. So feel free to contact me through my website. Thank you very much.